back to NEET Prep Go. So today we'll be again doing another chapter, biological classification, super fast. So without any further ado, let's get started. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use. So earlier the classification, there have been many attempts since the dawn of civilization for to classify living organisms, but it was done instinctively. There was no as such criteria that was scientific, but it was just born out of a need to use organisms for our own use for food, shelter and clothing. Aristotle was earliest to attempt a more scientific base of classification. He used morphological characters. He classified plants into trees, shrubs and herbs, and he divided animals into anima and inanima, that is those which had RBCs, inanima, and those which did not have RBCs, anima, that is blue-blooded or colorless blood, all that. Then came Carolus Linnaeus, who gave the two kingdom system of classification. Two kingdom system of classification had two kingdoms, that is Plantae and Animalia, and was developed that included plants and animals respectively. Then this system did not distinguish between the eukaryotes and the prokaryotes, unicellular and multicellular organisms, and photosynthetic and non-photosynthetic organisms. So what were the drawbacks of two kingdom classification given by Carolus Linnaeus? That is, it did not distinguish between multicellular and unicellular organisms, prokaryotic, eukaryotic organisms, and photosynthetic, non-photosynthetic organisms. So classification of organisms into plants and animals was done and was easy to understand, but a large number of organisms did not fall into either category. Therefore, the two kingdom classification which was used for a long time, was found inadequate. Besides gross morphology, there was a need to include other characteristics as well, such as cell structure, the nature of cell wall, the mode of nutrition, habitat, method of reproduction, and most importantly, evolutionary or phylogenetic relationships. That is relationships between the same ancestors, phylogenetic relationships that we saw in evolution. Classification systems for the living organism have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms can be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. Now let's see the five kingdoms: Monera, Protista, Fungi, Plantae, Animalia. Monera are prokaryotic, rest all Protista, Fungi, Plantae, Animalia are eukaryotic organisms. Difference between prokaryotic and eukaryotic, that is, prokaryotic organisms are not membrane bound. They do not have membrane bound organelles. Their nucleus, they have a nucleus, they do not have a proper membranous nucleus or nucleoplasm or anything. They have 70S ribosomes, whereas eukaryotic organisms have 70S plus 80S ribosomes. Then there are no cell organelles, that is such as Golgi apparatus and all. They are all absent. So basically primitive, that is, cell is very primitive, whereas eukaryotic cells are very complex. So they were primitive prokaryotes, non-cellulosic, that is, the cell wall was non-cellulosic. It was made up of polysaccharide and amino acid. In Protista, cell wall was present in some. That is such as the proteinaceous and euglena, we will see ahead, called as pellicle. Now fungi, that is chitinous cell wall is present, that is, cell wall has chitin major component as well as hemicellulose and all, pectin and all. Then Plantae cellulosic wall. In Animalia, cell wall is absent. The nuclear membrane, like I said, nuclear membrane is absent in Monera. In Protista, Fungi, Plantae, Animalia, it is present because they are all eukaryotic. Then Monera is cellular, that is unicellular body organization. Protista also is unicellular, so Monera are unicellular prokaryotes, Protista are unicellular eukaryotes. Fungi, Plantae and Animalia all are multicellular organisms. In fungi there is multicellular and loose tissue. In Plantae, tissue organ. In Animalia, tissue organ as well as organ system. There is a lot of many organs combined to form your, such as pancreas, large intestine, small intestine, stomach, all of them combine to form your digestive system, just like that. Autotrophic and liver and all. Monera. Autotrophic, the Monera mode of nutrition. Autotrophic, that is they can make their own food, either chemosynthetic autotrophic, that is using oxidation of chemicals, or photosynthetic autotrophic, that is by the sunlight, that is, and chlorophyll, that is the way plants make. And also heterotrophic, that is do not produce their own food, they depend on others for food and nutrient. Heterotrophic, and heterotrophic, they can be saprophytic, that is feeding on dead and decaying matter, as well as parasitic, that is feeding on a host. Then protista can be autotrophic, that is photosynthetic, as well as heterotrophic. Then fungi, they are only heterotrophic, they are not phototrophic, they are only heterotrophic, parasitic, and saprophytic, but majorly saprophytic. Then plantae, autotrophic, photosynthetic. Plants are not heterotrophic, they always produce their own food, and they are photosynthetic. Exception are Venus flytrap and Rafflesia and all. Then Animalia, heterotrophic. Again, animals, they'll never be autotrophic. That too holozoic, like we eat. That is, we convert complex substances, break down into simple, and then for absorption of our absorption, and saprophytic, that is on dead and decaying matter. So first, Aristotle, he divided plants into herbs, shrubs, and trees, and animals into anima without RBCs, anima with RBCs. Then came two kingdom classification by Carolus Linnaeus, who gave Plantae and Animalia. Then came three kingdom classification by Ernst Haeckel. What did he do? Uh, he gave three kingdom classification, that is, he added a third kingdom, Protista. Then came four kingdom classification, that is given by Herbert F. Copeland. What did he do? He added Monera. And then lastly came your five kingdom classification, which was given by R.H. Whittaker in 1969. The kingdoms were Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him included cell structure, then body organization, mode of nutrition, reproduction, and phylogenetic relationships. The three domain system also has been proposed, which divides kingdom Monera into two domains, that is Archaea, Eukarya, that is Archaea, bacteria, and the rest are Eukarya. Three domain system, that is Archaea, Eukarya, Archaea, bacteria. Archaea bacteria, that is both of them divided by that is three domain system, that is two domains, Archaea and Eukarya, made by Monera only, dividing Monera into two domains, and the third is Eukarya, leaving the eukaryotic kingdoms in the third domain, thereby a sixth kingdom classification. You will learn that this system is in higher classes. Let us look at this five kingdom classification to understand the issues and considerations that influence the classification system. So, in earlier classification system, bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, and angiosperm were under plants. So, your bacteria, that is cyanobacteria, blue-green algae, then your archaebacteria, fungi, mosses, fern, gymnosperm, and angiosperm, all of them were under plants. The character that unified this whole kingdom was that all organisms had a cell wall in their cells. So basically, in two kingdom classification, Carolus Linnaeus, what did he base the two kingdom classification on? That is on the presence of cell wall. That is, all those which had cell wall were put in plants, and all those which did not have cell wall were put in animals. Then this placed together groups which widely differed in their characteristics. That is, it brought the prokaryotic bacteria and the that is the blue-green algae or cyanobacteria with other groups which were all eukaryotic. Then it grouped the unicellular organisms and the multicellular organisms, such as Chlamydomonas, Pyrogyra, which were placed under algae. That is, the classification did not differentiate between heterotrophic group, that is fungi, and autotrophic green plants. Then it showed a characteristic difference in the cell wall composition, that is fungi had chitin whereas green plants had cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom called as kingdom fungi. All prokaryotic organisms were grouped under kingdom Monera. Unicellular eukaryotic organisms were in kingdom Protista. Kingdom Protista had brought together Chlamydomonas, Chlorella, which was earlier placed in algae within plants, both having cell wall, and Paramecium and amoeba, which did not have cell wall and showed predator kind of instinct, that is pathogenic, earlier placed in the animal kingdom together in Protesta. It has put together organisms which were placed in different kingdoms. This happened because the criteria for classification changed, and this kind of change can happen in future also, depending on the improvement in our understanding of characteristics and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only in the morphological, physiological, and reproductive similarities, but also phylogenetic based on the evolutionary relationships. That is, if two have common ancestors, then they will be placed together in the same group. So phylogenetic or evolutionary relationships are also very important. In this chapter, we will study characteristics of kingdom Monera, Protista fungi. Of the Whittaker system classification, the kingdoms Plantae and Animalia are commonly referred to as plant and animal kingdoms. Now kingdom Monera. So bacteria are the sole members of kingdom Monera. They are the most abundant microorganisms. So which are the sole members of kingdom Monera? Bacteria. Bacteria are the sole members of kingdom Monera. They are most abundant microorganism. Which are the most abundant microorganism? Bacteria. Bacteria are the sole members of kingdom Monera, and they are the most abundant microorganism. They occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They can also live in extreme habitats such as hot springs called as thermoacidophiles. Then अच्छा वो नीचे आता है या सॉरी it will come in archibacteria, sorry. Hot spring, desert, snow, deep oceans, in many places where few or no life forms can survive, there bacteria will survive. So many of them live in or on other animals as parasites. Living in other animals is endoparasite, and on is ectoparasites. Bacteria are grouped under four categories based on their shape. First is rod-shaped bacteria, Bacillus, plural is Bacilli. Then spherical-shaped bacteria, Coccus, plural is Cocci. Then comma-shaped bacteria, Vibrium, plural is Vibrio. And spiral-shaped bacteria, Spirillum, plural is Spirilla. So this is spherical-shaped cocci. This is rod-shaped bacilli. We having spore. Then this is spirillum, spiral shaped, having flagellum, and vibrio or comma shaped. The bacteria lack cilia, by the way. They do not have cilia. They have flagella, but they do not have cilia. They have pili for conjugation, but they do not have cilia. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most metabolic diversity. That is, who shows the most extensive metabolic diversity? Bacteria. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates, whereas most of the bacteria, they can be photosynthetic autotrophic or chemosynthetic autotrophic, whereas vast majority of bacteria are heterotroph. They depend on other organisms and or on dead organic matter for food. Archaebacteria. Archaebacteria are special. Why? They live in some of the most harsh habitats, such as extreme salty areas, halophiles, or hot spring thermoacidophiles, or marshy areas, methanogens. So archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. So why can archaebacteria compared to other bacteria survive in extreme conditions and are special or different or have some unique characters? Because they have, that unique character is they have different cell wall composition. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane or biogas from the dung of these animals. Then eubacteria. There are thousands of different eubacteria or true bacteria. They are categorized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria are also called as blue-green algae. They have chlorophyll A similar to green plants, and they are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called as heterocysts. So eubacteria, there are thousands of different eubacteria, also called as true bacteria. They are categorized by the presence of a very rigid cell wall. Then if they are motile, then they will have a flagellum. The cyanobacteria are also called as blue-green algae. They have chlorophyll A, which is similar to green plants, and the blue-green algae or cyanobacteria, they are photosynthetic autotrophs. So they can synthesize their own food by performing photosynthesis. Cyanobacteria are unicellular, and they can be colonial or filamentous or freshwater or marine or terrestrial algae. Colonies are surrounded by a gelatinous sheath, and they form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. So some of these organisms, that is of eubacteria, blue-green algae, photosynthetic autotrophs, or cyanobacteria, some of them can fix nitrogen in specialized cells which are called as heterocysts. Example are nostoc and anabana. Then chemosynthetic autotrophic bacteria, they oxidize various inorganic substances such as nitrates, nitrites, and ammonia. Chemosynthetic autotrophic bacteria, they oxidize various inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. So eubacteria or true bacteria, they are categorized by the presence of a rigid cell wall. If motile, a flagellum, and if they are motile, then they have a flagellum. Cyanobacteria is also referred to as blue-green algae, and they have chlorophyll A, which is similar to green plants. So they are photosynthetic autotrophs. They are unicellular, colonial, or filamentous. They can be freshwater, marine, or terrestrial. Their colonies are surrounded by a gelatinous sheath, and they form blooms in polluted water bodies. Some of them can fix atmospheric nitrogen in specialized cells called as heterocysts, and the examples are nostoc and anabana. Then chemotrophic bacteria, sorry, chemosynthetic or chemotrophic bacteria, chemoautotrophic bacteria, that is they um, oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for ATP production. They play a great role in recycling nutrients such as nitrogen, phosphorus, iron, and sulfur. What are the nutrients that the chemosynthetic autotrophic bacteria they oxidize? They recycle nitrogen, phosphorus, iron, and sulfur. Nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant. They're decomposers. They have significant impact on human affairs. They help in making curd from milk, that is your lactobacillus, production of antibiotics, then fixing nitrogen in legume roots, such as your rhizobium. Some are pathogens causing damage to human beings, uh, such as your Haemophilus influenza, Streptococcus pneumoniae. Then crops also they'll cause harm. Then farm animals and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to another. That is conjugation. They mainly reproduce by fission, but under unfavorable condition they produce spores. So mainly asexual reproduction fission takes place, but under unfavorable condition they produce spores. They can also produce by sort of sexual reproduction by adapting a primitive type of DNA transfer from one bacterium to another. Mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and they can survive without oxygen. So there are so many important points about mycoplasma. Firstly, they do not have a cell wall. They lack a cell wall. Secondly, they are the smallest living cells known. And thirdly, they can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Now we come to kingdom Protista. All single-celled eukaryotes are placed under kingdom Protista, but boundaries of this kingdom are not well defined. What may be a photosynthetic protestant to one biologist may be a plant to another. Such as, Chlamydomonas, chlorella and all. In chlorophycy also, you will see them coming as examples. So what would be what would be a photosynthetic protestant to one biologist? It can be a plant to another biologist. So the boundaries of this kingdom are not that well defined. Then in this book we include chrysophyte, dinoflagellate, euglenoids, protozoans, and slime molds. Members of Protista, they are primarily aquatic. Then the kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryote, protestant cell body contains well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. So cilia is present in Protista. Protists reproduce asexually and sexually by process involving cell fusion and zygote formation. Chrysophytes. Chrysophytes include diatom and golden algae, also called as desmids. They are found in freshwater as well as in marine environment. They are microscopic and float passively in water currents, plankton. Most of them are photosynthetic. In diatom, cell wall are made up of two overlapping shells which fit together like in a soap box. The walls are embedded with silica and thus these walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat, and this accumulation over billions of years is referred to as diatomaceous earth. And being gritty, this soil is used for polishing, filtration of oils and syrups. Diatoms are the chief producers of ocean. Then dinoflagellates. These are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigment present in their cells. The cell wall have stiff cellulosic plates on the outer surface and most of them have two flagella, one transverse and one longitudinal. Transverse one is a furrow between the wall plates. Very often the red dinoflagellates are called as goniaulacs. They undergo rapid multiplication and because they undergo rapid multiplication they make the sea appear red, and toxins released by such large numbers may even kill other marine animals such as fishes. The euglenoids. Euglenoid are freshwater organisms. They are found in stagnant water. Instead of a cell wall, they have a proteinaceous rich layer called as pellicle, which makes their body flexible. They have two flagella. One is short, another one is long. Though they are photosynthetic in the presence of sunlight, when unfavorable condition arises and they are deprived of sunlight, they behave like heterotrophs. They are predating on other small organisms. Interestingly, the pigments of euglenoids are identical to those which are present in higher plants. Example is Euglena. Then slime molds. Slime molds are saprophytic protists, that is, they feed on dead and decaying matter. Then their body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form an aggregation which is called as plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates to bear fruiting bodies at their tips, fruiting bodies bearing spores at their tips, and these spores possess true walls. They are extremely resistant and can survive for many years under adverse conditions. The spores are dispersed by air currents. Then protozoan. All protozoans are heterotroph and live as predators or parasites. Then they are believed to be primitive relatives of animals, and there are four major groups of protozoans. First is amoeboid protozoan, then flagellated, then ciliated, and last is sporozoan. So firstly, amoeboid protozoan, that is, they live in fresh water, seawater, and moist soil. They move and capture their prey with the help of their false feet or pseudopodia. Then many marine forms have silica shells on their surface. So marine forms of amoeboid protozoans have silica shells on their surface, and some of them, such as Entamoeba histolica, causes amoebiasis. That is, it is a parasite and it causes amoebiasis in human beings. That is your abdominal pain and diarrhea and all. Then flagellated protozoans. Flagellated protozoans, that is, the members of flagellated protozoans are either free living or parasitic. They have flagella. The parasitic form causes diseases such as sleeping sickness, which is caused by which flagellated protozoan? Trypanosoma. Then ciliated protozoans. Ciliated protozoa are actively moving aquatic organism because of the presence of thousands of cilia on their surface, so which helps them in locomotion or movement. They have a cavity gullet which opens to the outside of the cell surface, and coordinated movement of the rows of cilia causes the water-laden food to be steered into the gullet. So let's say this is a ciliated protozoan. Then they have so many cilia, and here there's the gullet. So because of this cilia, the water laden with food, it steers into the gullet. Then example is paramecium. Then sporozoans, diverse organisms which have an infectious spore-like stage in their life cycle. The most notorious is plasmodium. There are four uh, of plasmodium. Do you know four variants of plasmodium? That is, the most common one which happens to everyone is plasmodium vivax. Then there is plasmodium malariae. Then the most bad is malignant malaria, which is caused by plasmodium falciparum, which has very serious effects and can even lead to death. A disease with staggering effect on human population. Then we come to kingdom fungi. The kingdom fungi constitute unique kingdom of heterotrophic organisms. Yes, they show great diversity in morphology and habitat. You must have seen fungi growing on moist bread and rotten fruit, the common mushroom which you eat, and the toadstools. They are also fungi. White spots which are seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Yeah, we had seen yeast, that is Saccharomyces cerevisiae. It is also called as baker's yeast, used to make bread, pastry, and all. It is also called as dough and all. It is also called as brewer's yeast, used to make beverages. Then the white spots seen on mustard leaves are due to a parasitic fungus. Think Puccinia. Sorry, that is wheat rust causing Puccinia, huh? Some are the source of antibiotics, example penicillin, which was discovered by Alexander Fleming. Then fungi are cosmopolitan, that is, they can occur everywhere: water, air, soil, animal, plants. Then they prefer to grow in warm and humid places. Uh, Food is kept in refrigerator to prevent it from going bad due to bacterial and fungal infections. Then because if it is in the moist condition, or that is, it should rather be in a very cold and dry condition than being in a moist and wet condition outside. With the exception of yeast, which are unicellular, all the fungi are filamentous, multicellular. Their bodies consist of long, slender, thread-like structures called as hyphae. Long, slender, thread-like structures called hyphae. The network of hyphae, that is the mesh-like network of hyphae, it is called as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm syncytium, and these are called as coinocytic hyphae. Others have septae or cross walls, and in their hyphae. Then the cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic. They absorb soluble organic matter from dead substrates, and hence they are called as saprophytes. Those that depend on living plants and animals are called as parasites. They can also live as symbionts, that is in association with algae as lichens or with the roots of higher plants such as mycorrhiza. Then reproduction in fungi can take place by vegetative means, fragmentation, fission, budding. Asexual reproduction is by spores called as conidia or sporangiospores or zoospores. So reproduction in fungi can either take place by vegetative means, that is vegetative reproduction by fragmentation, budding and fission, or it can take place by asexual reproduction by spores. What are the asexual spores? Asexual spores are conidia, sporangiospores and zoospore. CSZ and oospore, ascospore, basidiospore. OAB. Oospore, ascospore, basidiospore for sexual reproduction, sexual spores. Various spores are produced in distinct structures. These are spores are produced in fruiting bodies. Then what does the sexual cycle include? It includes the fusion of protoplasm between two motile or non-motile gametes called as plasmogamy, fusion of the two nuclei karyogamy, and meiosis in the zygote, which results in haploid spores. Meiosis in the zygote, which results in haploid spores. So first plasmogamy, then karyogamy, then meiosis in fungi. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately results in the formation of diploid cell. However, in other fungi such as Ascomycetes and Basidiomycetes, an intervening dikaryotic stage, that is two nuclei per cell, occurs. So in most of the cells, that is two haploid hyphae, that is they come together of compatible mating types and they fuse, and the fusion of the two haploid cells result in the diploid cell in most of the fungi, but in some others such as example Ascomycetes and Basidiomycetes, there is an intervening dikaryotic stage, that is two nuclei per cell called as dikaryon, and the phase is called as dikaryophase. Later the parental nuclei fuse and become diploid. The fungi form fruiting bodies in which the reduction division occurs. So after fusing the zygote, then the fruiting body is made, inside which the meiosis occurs, reductional division occurs, leading to the formation of haploid spores inside the fruiting bodies. Morphology of the mycelium made a mode of spore formation and fruiting bodies. What are the basis of division of kingdom into various classes? That is morphology of the mycelium, the mode of spore formation, and fruiting bodies. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. So the members of Phycomycetes, they are found either in aquatic habitats, on decaying wood, the members of Phycomycetes are found in aquatic habitats on decaying wood in moist and damp places or as obligate parasites on plant. The mycelium is aseptate, that is does not have cross walls, it is continuous, and coinocytic, continuous, multinucleated. Then asexual reproduction takes place in zoospores, which are motile, or by aplanospores, which are non-motile. So zoospores are motile, aplanospores are non-motile. These spores are endogenously produced in a sporangium. So zoospores, motile, aplanospores non-motile, they are in phycomycetes, they are endogenously produced in sporangium. A zygospore is formed by the fusion of two gametes. These gametes are similar, can be either similar in morphology isogamous or dissimilar and isogamous, or the male is small, female is large, that is oogamous. Some common examples are mucor, rhizopus, and albugo, which is the parasitic fungi on mustard. The parasitic fungi on mustard is albugo. And Puccinia causes rust of wheat. And parasitic fungus on the bread mold is rhizopus, and the parasitic fungus on mustard is albugo. And albugo, mucor, and rhizopus, all three of them come under phycomycetes. Then ascomycetes, or commonly called as sac fungi. Ascomycetes are mostly multicellular, example penicillium, or they can be rarely unicellular, example yeast, Saccharomyces. They are saprophytic, decomposers, parasitic, or coprophilous, that is growing on dung. So commonly ascomycetes are called as sac fungi. They are mostly multicellular, penicillium, or they can be rarely unicellular, such as yeast, penicillium yeast. Then they can be saprophytic, that is feeding on dead and decaying matter, decomposers. Then parasitic, living on other host, and or coprophilous, that is growing on dung. Mycelium is branched and septate, has cross walls, and it is branched. Then asexual spores are conidia, which are produced exogenously on special mycelium called conidiophores. So in ascomycetes, in ascomycetes, the in phycomycetes there were motile zoospore and non-motile aplanospore, which were produced endogenously under sporangium. Here in ascomycetes there is conidia, which are asexual spores, and they are exogenously produced on conidiophores. And conidia on germination produce mycelium. Sexual spores are called as ascospores, these are endogenously produced in sac like asci, and they are so in ascomycetes there is asexual spores conidia, which are exogenously produced on conidiophores, and there is sexual spores ascospores, which are endogenously produced in sac like asci. The asci are arranged in different types of rooting bodies called as ascocarps. Some examples are aspergillus, claviceps, neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. So claviceps, aspergillus, claviceps, and neurospora. These three are the examples, and also penicillin, penicillium, and yeast. They are all examples of ascomycetes. And neurospora is used extensively in biochemical and genetic work, and many members like morels and truffles are edible and they are considered as delicacies. Basidiomycetes, commonly known forms of basidiomycetes are mushroom, bracket fungi, or puffball. They grow on soil, on logs, tree stump, in living plant bodies as parasites. That is example rust and smut. The mycelium is branched and septate, just like ascomycetes. Asexual spores are not found, but vegetative reproduction is by fragmentation. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains of genotype. So asexual spores are not there, sex organs are absent, vegetative reproduction is there, that is fragmentation, but plasmogamy is brought about by two vegetative or somatic cells of different strains or genotype. That is, their protoplasm fuse and plasmogamy occurs. The result is a dikaryotic stage, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. So basidium fruiting body inside that, that is basidiophore. Basidiospores are formed after karyogamy and meiosis, and these basidiospores, they are exogenously produced on the basidium. The basid or basidia, okay. This is the zygote. Basidium is the zygote, okay. The basidia are arranged in fruiting bodies called as basidiocarps. Some common members are agaricus, which is mushroom, Ustilago smut, and Puccinia, rust. Agaricus, Ustilago, and Puccinia. Then Deuteromycetes, Alternaria coletotrichum trichoderma, you must be knowing, commonly known as imperfect fungi. Only the asexual or vegetative phases are known. Sexual forms of the fungi were discovered and they were moved to other classes where they rightly belong to. It is possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage have been given another name and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of the members of Deuteromycetes were discovered, they were moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores called as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while in large numbers some of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Coletotrichum, and Trichoderma. Then kingdom Plantae and Animalia, and already we know. It includes all eukaryotic chlorophyll containing organisms called plants. Few members are particularly heterotrophic insectivorous plants such as your Venus flytrap, bladderwort, pitcher plant, and parasitic example Cuscuta. Then they have eukaryotic structure, and these are the exceptions. They have eukaryotic structure, prominent chloroplast, cell wall mainly made up of nucleus. Life cycle of the plants has two distinct phases: diploid sporophyte and haploid gametophytic stage. So this is your haploid gametophyte, which produces gametes, which are also haploid, and then they fuse to form your diploid, that is they fuse to form zygote, which develops to form your diploid sporophyte. Then this diploid sporophyte undergoes meiosis to form haploid spores, and then these spores form your haploid gametophyte by mitosis, and these alternate with each other. So if the part which is green, photosynthetic, and stays longer in a plant, that is whether between gametophyte and sporophyte, then that becomes the dominant stage. So in algae, that is chlorophyce, rhodophyce, phaeophyce, rhodophyce, green algae, brown algae, and red algae, the zygote is 2N is formed. From the zygote directly meiosis takes place, zygotic meiosis takes place, that we say, and spores are formed. So therefore they are haploid, that is their parent plant body is main plant body is haploid in algae. Then in bryophytes and pteridophytes, gymnosperm, angiosperm, zygotic mitosis take place. That is from zygote, that is by mitosis, that is same 2N diploid sporophyte is formed. Then so that is, since zygotic mitosis takes place, they are also called as embryophyta. Then in bryophytes also the main plant body is uh, haploid. Then in pteridophyte, that is gymnosperm, angiosperm, it is diploid. In pteridophyte though, there is haplodiplontic cycle, whereas gymnosperm, angiosperm diplontic, and these two is haplontic. Bryophyte and algae, they alternate with each other, saprophytic and gametophytic. There is alternate the length of them, whether these phases are free living or dependent on others, vary among different groups. Phenomenon is called as alternation of generation. Kingdom Animalia, by heterotrophic eukaryotic organism, multicellular, do not have cell wall, directly or indirectly dependent on plants for food. That is, if a deer is eating plants, it is dependent directly on food. If a lion is eating the deer, then the deer, that is, he is indirectly dependent on plants. Then they digest their food in an internal cavity and store reserves as glycogen or fat. Their mode of nutrition is holozoic, by ingestion of food, follow definite growth patterns, grow into adults, have definite shape and size. Higher forms have elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion, that is movement. They are motile in nature. Sexual reproduction is by copulation, that is of male and female, followed by embryological development. Population mating. Virus, viroid, prions, and lichens. Five kingdom classification of Whittaker. There is no mention of lichens and some acellular organisms such as virus, viroid, prion. They are briefly introduced here. All of those who have suffered the ill effects of common cold or flu know what effects viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification. They are not exactly living. They are not truly living. Living organisms are those which have a cell structure. Viruses are non-cellular organisms, do not have a cellular structure. They are characterized by having an inert crystalline structure outside the living cell. They are inert outside the living cell, that is, they are not really considered living. They do not show any signs of being living outside the host cell, but once they get into the host cell, they use the host mechanism to divide, replicate themselves, and eventually end up harming, killing the host. Yeah, so they basically head, collar, tail, sheath, plate, prongs, tobacco mosaic virus, PMV, bacteriophage. Okay, so once they infect the cell, they take over the machinery of the host to replicate themselves, killing the host. So will they be living or true living? So truly living or non-living? Don't know. Virus means venom or poisonous fluid. Who said this? Dmitry Ivanovsky in 1892. He recognized certain microbes as casual organism of the mosaic disease of tobacco. He used bacteria-proof filters, and they passed through. That is. He then found out that they are smaller than bacteria. Then in 1898, M.W. Bjornik demonstrated the extract of infected plants of tobacco could cause infection in healthy plants, and named the new pathogen virus and called the fluid Contagium vivum fluidum, infectious living fluid. Then W.M. Stanley in 1935 showed that viruses could be crystallized, and crystals consist of largely of proteins, inert and outside the specific host cell. They are obligate parasites. In addition to proteins, viruses also contain genetic material. It can be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein, and the genetic material is infectious. Viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. So the viruses which infect plants, they have only single-stranded RNA, whereas the viruses that infect animals, they can have single-stranded RNA or double-stranded RNA or and double-stranded DNA. Bacterial viruses or bacteriophages are the viruses which infect the bacteria. They are usually double-stranded. The protein coat called capsid is made up of small subunits called as capsomere, which protects the nucleic acid. in general, viruses that infect plants, they have a single-stranded RNA, whereas viruses which infect animals have a single-stranded RNA, double-stranded RNA, and double-stranded DNA. Can have. No virus has both RNA and DNA. Then bacterial viruses or bacteriophages are the viruses which infect bacteria. They are usually double-stranded. The bacteriophage is double-stranded. Bacteriophage is double-stranded. This is important. Bacteriophage is double-stranded. Then the protein coat is called as capsid. It is made up of many small subunits called as capsomere, which protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. That is, they are helically arranged in tobacco mosaic virus TMV. You saw right now that is like this, and polyhedrally arranged in bacteriophage. Then viruses can cause disease like mumps, smallpox, herpes, influenza. AIDS is in humans is also caused by a virus. Which virus? Human immunodeficiency virus. Then in plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing, yellow mosaic virus, vein clearing, dwarfing, stunted growth. Then viruses. In 1971, Theo Diner discovered a new infectious agent which was smaller in size than viruses and caused potato spindle tuber disease. It was found to be a free RNA and it lacked protein code, and that is found in viruses, and therefore he named it viroid. So viroid, that is in 1971, T.O. Diner, he discovered a new infectious agent which was smaller in size than viruses, and it consisted of free RNA, no protein code, and hence he named it viroid. What did it cause? It caused potato spindle tuber disease. The RNA of viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called as prions. The most notable diseases caused by prions are bovine spongiform encephalopathy (BSE), called as mad cow disease in cattle, and the analogous variant, that is those which eat the cattle, that is the beef, containing this virus, that is and has not been cooked very well, that is this BSE, bovine spongiform encephalopathy. Then they will get the variant CR Jacob disease in humans, and another Kuru disease is also caused by prions. Another disease called Kuru disease. Lichens are symbiotic association, that is mutually useful associations between algae and fungi. Algal component is called as phycobiont and fungal component is called as mycobiont. The algal component is obviously autotrophic, it gives food and nutrition to the fungal component, and fungal component is heterotrophic, depends on the algae. Algae prepare food for fungi, fungi provide shelter and absorb mineral nutrients and water for the algae. So close is their relationship that if one saw a lichen in nature, one would never imagine that it had two different organisms within them. Lichens are very good pollution indicators, they do not grow in polluted areas. So if you view a lichen and you saw that it is not growing properly, it disappears, then you will realize that pollution is very high. Wherever you see a lot of lichen, it means the pollution is low over there. So let's see all the examples once again quickly. That is example of Monera. Example of kingdom Monera, that is your lactobacillus, then blue-green algae, cyanobacteria, in that nostoganobacteria and nitrogen fixers, then your chemoautotrophic bacteria, then your when chemoautotrophic you can have that your sulfur bacteria, that is then heterotrophic bacteria, lactobacillus, um, your rhizobium fixing nitrogen in legume roots of plants, then mycoplasma, lac cell wall, Then your typhoid, that is Salmonella typhi. Then your Vibrio cholerae. Then tetanus, Clostridium tetani. Then your citrus canker, the virus, sorry, the bacteria called citrus canker. Okay, that much. Pneumonia, also Streptococcus pneumoniae, Haemophilus influenza, all those. Then Protista, Chrysophytes, example, diatoms, golden algae, that is desmids. Then dinoflagellates, example is Gonia olac, which causes red type, red sea. Then euglenoids, example is euglena. It has a proteinaceous layer called pellicle insulatum cell wall, which makes it flexible. Then slime mold. Then protozoan, that is amoeboid protozoan, entamoeba histelica, flagellate protozoan, trypanosoma causing cereal sickness, entamoeba histelica, producing amoebiasis, causing amoebiasis, and ciliated protozoan is paramecium, and sporozoan is plasmodium, which causes malaria. Malignant malaria is plasmodium falciparum. Then kingdom fungi, Phycomycetes, example is Bucor. Then albugo, which is a parasitic fungus on mustard. Then rhizopus, which is a bread fungi. Then Ascomycetes, then is your truffles and morels, which are delicacies and eaten by many people. Then second example is your claviceps and neurospora. That is, neurospora is used extensively in biochemical and genetic work. And Aspergillus niger, which produces citric acid. And also your penicillium, that is penicillin, antibiotic penicillium, and your yeast, Saccharomyces cerevisiae. So what are the examples of Ascomycetes? That is, your yeast is an Ascomycete, penicillium is an Ascomycete, claviceps, neurospora, Aspergillus, then your truffles and morels. Basidiomycetes example is what you love to eat, that is agaricus mushroom. Then Ustilago, smut, rust and smut, right? So Ustilago. And the one that causes rust of wheat, that is Puccinia. So rust and smut come in Basidiomycetes. The rust is Puccinia, which causes rust of wheat, and smut is Ustilago, and your common eating mushroom. Deuteromycetes example is Alternaria, Colitotrichum, and Alternaria, Colitotrichum, and Trichoderma. Then virus causes your tobacco-mediated virus is an example. Then bacteriophage is an example. Then your rhinovirus, retrovirus, HIV/AIDS, they are all examples. Yellow mosaic virus. In plants, that is single-stranded RNA causing diseases. In animals, single-stranded RNA, double-stranded RNA, and double-stranded DNA, all of them cause diseases. Now, viroids, example is potato spindle tuber disease. Prions, example is mad cow disease, that is also called as bovine spongiform encephalopathy, BSE, and its analogous variant in humans is CR Jacobs disease. And also another example is Kuru disease. Then prions are abnormally folded proteins. They are similar size as viruses. Viroids are free RNA, that is no protein coat, and low molecular weight RNA, as well as lesser in size, smaller in size than viruses, and may cause potato spindle tumor disease. Firstly, Dmitry Ivanovsky bacterial filter, it passed through, then that is virus passed through the bacterial filter that is smaller than bacteria, he realized, and something else he realized. Then secondly, after Dmitry Ivanovsky, M.W. Bjornik, he said contagium vivum, contagium vivum fluidum, that is, he demonstrated that extract of the infected plant of tobacco could cause infection in new plant. Then W. M. Stanley in 1935, he crystallized the virus and showed the crystalline form of virus. Then largely they consist of proteins and they are inert outside. Deodiner in 1971 discovered thyroid which caused potato spindle tumor disease. That's it. Thank you so much.